We always start with our mission, which is to cure Hao-Fountain syndrome by funding research and identifying more patients. And we are really making strides toward this effort. And I would like you to meet everybody else on the call. I will go through the list as I see it and have people introduce themselves. Bo, would you like to introduce yourself, or I can, since you're in the car?
You can.
Okay. I think everybody knows Bo. He founded Hao-Fountain along with his wife when his beautiful daughter Tess was diagnosed. Mark.
Well, hi, Mark Verdilla, Research Program Manager, Foundation for Prader-Willi Research. Prader-Willi syndrome, most of the patients are missing a gene called MAGEL2, which you'll probably see in some of Larry's schematics, also plays in some of the same games as USP7. So our foundations are looking at a lot of the same processes. And so the Prader-Willi syndrome community is also very interested in progress with the Hao-Fountain community.
Wonderful. Our dear Irina.
Everybody, my name is Irina Basanova. I'm at the University of Connecticut Health Center. I'm a structural biologist. My lab studies USP7 and its mutations that are affected, that are present in Hao-Fountain syndrome. So welcome.
Tara.
Hey everyone, I'm Tara Bayat. I work with Dr. Clementina Fontasser. I'm a PhD student at Texas Tech University. I'm mostly working on the role of MAGEL2 in the regulation of mRNA translation in a MAGEL2 knockout mouse model in Prader-Willi and Hao-Fountain syndrome. And I'm happy we joined this community of USP7, and hopefully we do more.
Dennis.
Hello, I'm Dennis. I'm also a PhD student in Dr. Fontasser's lab, and I also work primarily on MAGEL2 and its binding partners and its role in transcription.
Isabella.
Hi, everyone. I'm Isabella. I'm a PhD student in Sarah Perliger's lab here at Dana-Farber and Harvard Medical School. And I work on building chemical tools for studying USP7.
Mandon.
I'm feeling quite inadequate. I'm just a dad. I sell insurance. I have an insurance agency, and I understand a lot of that legal jargon. However, this science is tough to understand. But I'm doing my best, so don't ask me any complicated questions. But I thank all of you guys for being here. My son's name is Hendrix Foley. He's awesome, and he's one of the guys that are hoping that these meetings bring lots of knowledge and forward movement in the movement. So thank you for having me.
Clementina.
Hi, I'm Clementina Fontasser. I'm together with Tara and Dennis trying to help know more about what these proteins, MAGEL2 and its binding partners, do on cellular and molecular and physiological level. And I would like just to tell or comment on previous comment. I think you parents are doing much harder job that we do, and you are our inspiration. And Bo, I'm listening to your podcast, and you're great. So your children and parents are our inspiration for sure.
Wow, thanks.
Janelle.
Hey, so I'm Janelle Stanton. I am a current postdoc in University of Florida. Recently moved last year from University of Limerick, where I focused on USP7 using stem cell and organoid models. We still haven't gotten the paper out to the public yet, but we're working on it. I've done a lot of background in proteomics, transcriptomics, and I moved over to genomics here in UF Health. So.
And Emily.
Hi, I'm a graduate student at UConn Health for Dr. Betsanova, and I study really just Hao-Fountain syndrome. So I'm working on the effects of mutations on the USP7 function. I'm really just trying to understand better how the disease works, if we can do anything to help patients and all that.
We seriously do have the best community of patient families, scientists. The symposium last year felt more like a welcome family reunion. Like not the type of family reunion where you're just forced to go, like one that you seriously wanted to attend. And we are going to have another one this year. Becky, would you like to say some things about it?
Sure. We're so excited that this year we're going to have our symposium and our family conference September 18th and 19th, and it's going to be at the Hilton Garden Inn in Brentwood, which is just a little bit south of Nashville. We are finalizing everything for the hotel this week, and I'm going to get registration open hopefully by the end of this week. If not, it'll be next week. For scientists to register, for families to register, and it's going to include all of the important information for everybody. This year, we're going to have 2 days. The first day will be a scientific track. The second day will be more geared towards families. And of course, everybody is welcome to attend both days. We are going to do a few things differently this year as far as for parents with children. We're going to have a breakout room that'll have the conference streaming. So if you need to step out, there'll be a room where you can still watch and participate with your child with some toys or activities. And if anybody has questions, you can feel free to ask me right now and I'll hopefully try to answer them. And if that's it, I'll try to send everything out this week.
And like Clementina said, Bo's podcast is really fabulous. It's an appropriate length. He hits on every key issue, and he's also just a delight of a person. Funding opportunities, a little bleak right now with the NIH and CIRM not really issuing or paying awards, I guess. There are some opportunities through Uplifting Athletes, the American Epilepsy Society, and the Centers for Disease Control, although it didn't, I just realized the CDC falls under federal funding, so not totally sure. Our, oh my gosh, 25 priorities. Even though we're a month into 2025, it kind of feels like we're a year in. We are working on understanding the mechanism of disease and establishing correlations and hopefully biomarkers with much bigger communities like the cancer communities. We are working on 2 small molecule screens, one on the Novartis library and one on the Grand Library of FDA approved drugs. A disease concept study will be published. And we're also really looking at recruiting more medical doctors to own our patient group. We have so many opportunities for someone early in their career or looking for a rare disease to champion to work with our community. We have Dr. Schaff in Heidelberg, and he's wonderful, but the appointments are a little inconvenient for everybody in the US. And we are using the Matrix software here through Combined Brain. And what that means is we have patient surveys, patient data, medical records, all in a central location. And this information is accessible. And we have an IRB. And there are publication opportunities, the opportunity to expand a natural history study. Really, we can go anywhere with this. We're just trying to identify the right partners for us. Some articles were recently published. Hao's article coming from the Kim lab at WashU was formally published. We had a virtual lab meeting about this last year. And then similar for the molecular signature coming from Peter Henneman's lab. This year, we're going to be having our virtual monthly scientific meetings. If you have any ideas on something you would like to learn more about, or if you would like to present, please let us know. We are also hosting monthly events with the families and the patient community just for education. We would love to have your involvement on all of the above. And we're still working on consolidating resources. So adding plasmids to AdGene, working on our large Google Doc spreadsheet of all available resources. Again, if you have something, let me know. Irina, I feel like I reach out to you every so often to make sure that we're up to date. I should probably do that again.
Yeah, we actually deposited more than 30, close to 40 expression plasmids for USP7 wild type and various Hao-Fountain mutations. They are on gene right now.
No way, that's awesome, Irina. Oh, how cool. Okay.
Can I ask a science question about that?
Sure.
So what vectors are they in? Are they for like mammalian cell culture or?
No, they're for bacterial expression. So mostly for protein expression purification. We do have, if that's something you're interested in, we have, I believe, an expression system for mammalian cells for the full length USP7.
I'll give my talk and then you can tell me.
Okay, and then introducing Dr. Ryder. It's Ryder, right?
Mm-hmm.
From Tulane, and Dr. Ryder will be discussing using dental pulp stem cells to study neurogenetic diseases. I like to call him the tooth fairy. And Dr. Ryder, I will hand things over to you.
People have called me worse, so it's not such a bad one. All right. Well, I want to thank everybody for having me here for this meeting. It's the first time I've been here, but I do know some of you from the Prader-Willi field and, going to meetings and, it seems like we're on a common path here. And it was just a matter of time, I think, before I came to speak specifically to the USP7 community. So, yeah. So I guess you said it was sort of an informal thing, and there are some scientists here, some parents. So I didn't cram it full of slides. I didn't want a marathon, So I'm going to talk about what we're working on in general and how it relates to USP7, but I'm also open to you stopping me at any point and asking questions about any stage of it. So don't feel like you can't interrupt. I'm more than happy to take questions. Okay, so I just moved my lab. I was at the University of Tennessee Health Science Center for 19 years. And in August, I came to New Orleans and moved to Tulane. And I really love New Orleans, so we can talk about that later. And I love Tulane. The students are fantastic. The facilities are great. So this was a move up for me and my wife. So we're pretty happy about that. So I'm gonna talk in general, and then specifically, I'm going to talk more about Prader-Willi projects. But you'll see where USP7 fits into those projects. And then I might add some things that aren't even slides here because they're relevant. And it sounds like you guys have some reagents that could be useful too. So my lab is split into 3 parts, really. The first part we haven't worked as much on recently, but we have done studies on phenotypic analysis of duplication 15q syndrome. We've used a lot of human patient samples in the past, and so we do actually have stretched out into clinical work, but lately we have not. The rest of my lab is split between Drosophila models and what I'm going to talk about today, these stem cell models that we get from teeth. And I will briefly mention that the fly models we work on are also, very human disease specific. The number one thing we work on is screening for anti-epileptics using a model of duplication 15q epilepsy. And we've had some success in finding new compounds and even mechanism involved in epilepsy. But we're going to talk about teeth. So years ago, I was frustrated with the issue of not having access to neurons. I worked on, things like autism and epilepsy and really had no way to see what was going on in the actual neurons in individuals who had these syndromes. So we devised this plan. At the time, there were very few people who actually worked on dental pulp stem cells. And we looked around and found that there was maybe, I think at the time there was one lab that used dental pulp stem cells to make neurons. So we thought we would give it a shot. Okay. Now, what we developed in the interim there is that it really enabled us to collect a broader range of rare genetic disorders because of the way that we collect these stem cells. So it doesn't involve a clinic visit. It doesn't involve a blood draw. All that happens here is that if you have a syndrome on this list or one that we put on this list, and you want to contribute your child's tooth to our study, their tooth's going to fall out anyway, right? So typically people would just dispose of the tooth or the tooth fairy would take it or whatever, right? But we're actually using this resource and it's been great. So, and in fact, the response from the rare disease community has been fantastic, right? Because it enables you to actually directly contribute to research without leaving your house. So when the tooth falls out, you put it in this tube of media that we send you, and there's a little bit of documentation involved, a consent form and an SCQ, which is an assessment for autism that we can use remotely. So that you put all that stuff in the box, you send it back to us, and then we break open the tooth and grow stem cells from it. And then we typically freeze those stem cells back in our repository until we're ready to do specific studies. So our focus has been very heavy on chromosome 15, actually, for a variety of reasons, because we already worked on duplication 15q epilepsy. We've been working on Angelman syndrome for a while, and we started working on Prader-Willi syndrome maybe, oh God, it might be like 10 years ago now. And so if you look at our numbers, we actually have more samples from Dup15q, Prader-Willi, and Angelman than any of the other groups. And that's just intentional collection. So if you look at USP7, we've got 2 lines, at least on this slide. I think we do actually have just 2 lines, but we would love to expand that collection. And I will let you guys know how to do that. Unfortunately, I can't do it right now. I mean, I can't collect right now because I'm in the midst of getting the IRB approved at Tulane. And once I do, and I'll let you guys know, then it's just a matter of, in fact, I'm going to try and make it online so the parents can fill out the consent form online and we'll get notice and we'll get the genetics report and we will send out kits. So I would love to do that. I think that's a great direction for us to go here. But long and short of it is we have over 200 dental pulp stem cell lines from 14 different syndromes. And it's been very useful for us in a variety of ways, more than what I'm going to show you today. But we've been using them to make neurons, to make neuronal cultures that we can then do all kinds of different things with. And I'll show you just some of that today. So what do they look like? So, you know, the DPSCs themselves, the dental pulp stem cells, they kind of look like fibroblasts. They have this sort of flat morphology. Not very exciting to look at. They grow pretty well, but they will peter out. And we've actually, you know, done studies to show how many divisions we can get out of those stem cells. They're not immortalized. They're not de-differentiated from fibroblasts. They're not iPSCs. They have a limited division lifespan, right? And so far, we haven't really run out of any lines, but we do have some lines that grow better than other lines. And that is a limiting factor in the whole process. But once we do the differentiation, you see they start to change their morphology. They turn into these nice neuronal looking things with these processes. We actually have been very focused on some single cell work in these cultures in order for us to determine exactly what the collection of cell types is in there. And the preliminary work is really exciting because we see both excitatory and inhibitory neurons in the culture. We see astrocytes. We see things that we would expect to see in cortex, basically. So I think this is a really excellent model, better than a pure neuronal model, in my opinion, right? Because you need that support to really function. Neurons don't function in isolation, basically. All right, so this is for the scientists here. So how does this work? And this is going to be somewhat familiar to you guys who've been in the stem cell field. So there's an epigenetic reprogramming step. It's just a wiping of the methylation pattern in the DNA. There's neural induction with neurotrophic factors, and then neural maturation for about 4 to 5 weeks, typically. And then at that point, we do whatever we're going to do with them. They don't last forever as neurons. They don't, you know, the culture is limited in each experiment. So it's kind of a terminal process once we do this part. So they make things that you'd expect neurons to make, GABA A, MAP2. So you can see that in the DPSCs, neither of those markers are there. And then we differentiate into neurons and we see those markers. There's lots of markers. Lots of people have done lots of studies now showing that the appropriate markers are there, that these things are turning into neurons. We've actually shown that they have sodium, potassium, and calcium currents. Other labs have done this as well. So, you know, they walk like ducks, they talk like ducks. Okay, so I'm going to go over just a few things that we've been doing recently. And then I'm really happy to take questions too. So I'm excited to hear what you guys think. So, all right, so one of the studies I'll show you a little bit about is the mitochondrial study we published recently, well, a little bit time ago now, showing a decreased mitochondrial volume in neurons in Prader-Willi syndrome, but specifically in the UPD cases that have a positive ASD test by SCQ. So that's really important. So this is an ASD specific phenotype, autism specific. Then I'm going to focus on the circadian rhythm work we've been doing lately. Mark's really familiar with this. And we're making strides in this direction because we have some new equipment here at Tulane that's going to help us do some drug screening in this area. And then I can talk briefly about some collaborative studies that we've done. I don't know how much I harped on any of these individually, but you know, the truth is everybody who works on a rare disorder, you know, who knows about what we're doing tends to be excited. One of those people, Debbie Wiesmeyer at Lurie Children's Hospital, she and I have been collaborating for a few years now on two syndromes, actually, on one called ROHAD and one called central congenital hypoventilation syndrome. So she runs a clinic. She runs a autonomic disorder clinic and a sleep clinic. And so she's really interested in the molecular causes of, for example, CCHS, which is an extremely rare disorder. And she's seen pretty much all the kids, I think, that have this syndrome. So this is a case where, you know, you've got a really rare syndrome and we're able to still collect these important samples to study molecular and functional, neuronal functional experiments in this rare disorder. We've done some DPSC methylation studies that we published a few years ago with Janine LaSalle. That was really important work because it showed that the DPSCs looked more like embryonic stem cells than iPSCs. If you're talking about the whole methylome, the global methylome, that's an important point because that's going to imply that gene expression is probably more embryonic stem cell-like in these cells, and the potential for differentiation is different. And then we published some studies on endocytic recycling with Ryan Potts. That was a really good example of how you can use our system when you have an issue you want to address. We were more of a support role, but one of the things that we did that I love in that paper is the Prader-Willi deletion was his main interest, right? But the Angelman syndrome deletion, which is the reciprocal disorder, served as a really incredible control in those experiments because it looked just like neurotypical for those particular phenotypes that were MAGEL2 specific. That was important because we have those resources, so we could do those studies. And then we recently published a paper on piezo2 channels in Angelman syndrome, and that was really another example of where you could actually do electrophysiology. So they looked at this channel in Angelman syndrome neurons that we supplied to their lab. Okay, so let's talk a little about circadian rhythm. I guess I didn't talk about the ASD study, but that's all right. Some of you might know children with Prader-Willi syndrome have what's known as sort of daytime sleepiness. Schaff-Yang syndrome, which I already heard mentioned, is a related disorder related to PWS, and it's caused by mutations in MAGEL2, but 80% of the SYS kids have sleep disorders. That's really striking, right? So something's going on with sleep in both of these conditions, or quality of sleep. So we set out to use these DPSC neurons to look at circadian rhythm, something that I didn't even know was possible until we started these studies. And I collaborated with Andrew Liu at University of Florida, and he studies circadian rhythm in cell culture systems. So he helped us and gave us constructs for us to be able to look at the endogenous circadian patterns in these syndromes. So we're trying to figure out the molecular causes of the sleep issue, and I'll tell you how far we've gotten now and what we think is going on, and also how that involves USP7. And you know, we have a system that we're setting up in 96-well format for us to be able to screen for new drugs that specifically are treating the circadian rhythm defects that we found in PWS. So here's where it starts to get interesting to this group, I think. So I'm going to briefly go over these things. Some of you may know something about circadian rhythm. There's a feedback loop that happens. Essentially, what goes on is there's some transcription in the nucleus of Cry1 and PER2, right? And MAGEL2. These are all controlled in a circadian manner, right? And the genes that are important for driving that transcription are other clock genes, BMAL and clock, again, circadian cycling genes. And PER2 and CRY1 are involved in inhibiting this transcription, forming a feedback loop, right? So when you export CRY1 and PER2, there's an accumulation of CRY1 and PER2, setting you up for the next phase of the cycle. But that stability of that CRY1 protein and the PER2 protein are dependent on ubiquitin ligase activity. Specifically, this RBX1 SCF complex is the ubiquitin ligase that's going to mark these proteins for degradation, for cyclical degradation. However, USP7 is a deubiquitinase that stabilizes CRY1, right? Where does MAGEL2 fit in? Well, MAGEL2, as some of you alluded to earlier, interacts with USP7, and some of you are actually working on this. And so this is really important to us. So we kind of know that there's this literature out there that says that MAGEL2 and USP7 interact, but what are they doing, right? And what we think is going on is that MAGEL2 is fine-tuning the ability of USP7 to stabilize CRY1. So if you cause a defect in MAGEL2, for instance, in Hao-Fountain syndrome, and it's no longer able to stabilize CRY1, or it stabilizes CRY1 too much, we don't even know which direction we're going here, to be honest, you know, that can have a dramatic effect on the cycle, right? And now the cycle is affected that because CRY1 is not cycling normally, the circadian rhythm is not normal. And I'll show you what I mean by that. Oh, well, Hao-Fountain syndrome and Hao-Fountain syndrome, right? So that, you know, this is the way science is supposed to work, by the way. So we got involved in all of this because of daytime sleepiness in Prader-Willi syndrome, and now we're studying Hao-Fountain and Hao-Fountain syndrome, right? So the science has led us in the direction of studying all these other syndromes, not just because we wanted to, but because they affect the pathway that we've started to tease out. All right, so how do we do this stuff? So originally we did these studies, and most of the ones I'll show you, we've done in a device called the Lumicycler, which really isn't as easy to use as the way we're doing this now. But I'm going to run through the protocol just so you have an idea of what we're actually doing in the lab here. So Andrew supplied us with a PER2 luciferase vector. So what do I mean by that? Well, this vector has basically a PER2 promoter driving luciferase. So when PER2 is activated, luciferase is made. When PER2 is deactivated, there's less luciferase, or decrease in luciferase. So that promoter is the promoter itself is sensitive to this cycling in the cell, right? So this is a readout for normal circadian rhythm. So we put that vector into our dental pulp stem cells. We selected 4 cells that had that construct, expanded the DPSCs, differentiated them into neurons, synchronized them, and then put them in our Lumicycler. So that's this process here. We transduce them to select. We selected them, and then we differentiated them into neurons first for 4 weeks, and synchronized them with forskolin and IBMX, and that allows the entire culture to now synchronize its circadian rhythm together. Otherwise, you would just get sort of spaghetti, right? So now all the cells are synchronized to the same part of the cycle. And then we put them in there for 5 days. Now, the problem with that is the Lumicycler is going to detect the light coming off this luciferase construct, and that's fine, but the number of cells that actually are neurons that are making the signal is going to be variable among the cultures. And also, when we differentiate into neurons, it's not as clean as this image shows you. There are cells that die off during that process. So we don't know the total numbers, right? So we get signal and it is synchronized, but we can't look at amplitude. the intensity of that signal. Also, the cells are sealed in these plates and are not undergoing gas exchange. And so they don't do as well in the Lumicycle as we had expected or as we wanted. So I'll show you at the end how we solved that problem, I think. And we're going to do this a little bit differently in the future. So let's talk about circadian rhythms. Here is the rhythm in the dental pulp stem cells. Now, some of you in the iPSC field or just the stem cell field, embryonic stem cells, you know that you might know that there are actually no really distinct circadian rhythms in stem cells. And in fact, that's what we saw. There's not really a good rhythm among 3 different control individuals. We saw really no detectable rhythm. However, when we differentiate into neurons and we make these neuronal cultures and we mature them for 4 weeks, we put them in the Lumicycle and we get these beautiful patterns, right? So these individuals are not related. These are just 3 random neurotypical controls, and yet their rhythms are almost on top of each other, and they have consistent period lengths from peak to peak. Okay, so we were unsure whether we should use PER2 or BMAL initially. And that's because they're both clock genes and we had both constructs. So we tested them. And what's really nice about this figure is that PER2 and BMAL are oppositely cycling in the circadian cycle. And in fact, you can see that PER2 and BMAL oppositely cycle, right? When one's peaking, one's troughing, one's peaking, one's troughing. And that is the case for Prader-Willi dental pulp stem cell neurons as well. So at that point, we decided we would pick one and not use both of them. So we picked PER2, and the rest of the slides I'll be showing you are with a PER2 luciferase. Okay, well, let's get to the meat of the story. So initially, when we did these studies,
just to clarify,
Yes.
so these 2 proteins, the PER2 and BMAL, they function is to inhibit MAGEL2 production, or what was it?
Okay, so PER2 and BMAL are actually involved in transcription. Well, BMAL here binds to the E box with clock to drive PER2 transcription, right? So, and PER2 itself, the promoter for PER2, which isn't on here, is regulated in the same way, right? So these guys are in opposition, BMAL and PER2.
Thanks.
Does that make sense?
Yeah.
But we're not looking at the protein. We're using the promoter, driving luciferase. Is that clear? Or no? Yes? Okay. So if you look at the controls again, they're right on top of each other once again, and there's even more controls here. So they have a very consistent period length. Unfortunately, for a variety of reasons, I think the Lumicycle is not giving us the real period length, but it's very consistent from sample to sample. So it's a little longer than we would expect. It's 27 hours and not 24 hours. And I'll show you how that changes when we change the way that we treat these cells. When we looked at Prader-Willi, however, we did see a significantly shorter period length. So when we do the statistics, we could see that the control versus PWS, we saw a shorter period length, and then we saw some cell lines that actually had what we initially called a longer period length, but you can tell 27.4 versus 27.9 is really not significantly different. And there was a huge variance here. So we actually think that there's just a short length, and we haven't been able to fully characterize this longer period length, which may just be control period length. And why would that be? Because not everybody has sleep problems. So there are going to be individuals who match neurotypical controls in terms of endogenous circadian rhythm. So that's not a surprise. And I'll show you some data on Hao-Fountain, which might convince you that you know, those guys have a serious problem with their circadian rhythm. So we're focusing now on this short period length, and I just want you to sort of set your brain that way, because we think the long period length is really just maybe closer to control. All right, so as I said, we initially used these little plates that we had to seal with parafilm to put into the Lumicycler. And now we're switching to a 96 well plate format. We're still doing the same 4-week differentiation. We're still using per 2 luciferase. But now that we have it in a 96 well plate format and we're using a plate reader, we're able to do titrations of drugs like longdazin, which I'm going to show you in a second. And in fact, we're able to screen a lot more cells using a lot more compounds. So what are we actually measuring? I mean, I should have shown you this before, I guess, but you know, we're measuring the signal, the light that's coming off that luciferase construct and how it's changing over time. Okay. So this data is hot off the presses. Actually, this paper's back to review right now. So the lines don't look as pretty, but trust me, this data is really striking, and it's working the way we kind of expected in the plate reader. It's just a different system, so we can't sample the same way we were sampling in the Lumicycle. We're going to try and adjust some of those conditions soon, I hope. But what I want you to pay attention to here is if you look at B and you look at the untreated cells, now the cycle is closer, so the actual period length is closer to 24 hours in the controls. When we add a little bit of longdazin, so 0.5 micromolar, we do not see a significant increase in the period length, but when we get up to 1 micromolar, we do. So we can significantly increase the control period length using this drug longdazin, which is actually used to treat people with conditions related to circadian defects. When we look at the Prader-Willi line, this line 148, this line has a short period length. So if you look at, this is going to be 4 replicate samples from that line, and you look at control versus PWS 148, you see a significant decrease in period length. And then this is actually more striking than that other data that I showed you. So normal control was around 24, 20, 25 hours, something just under 25. And then these PWS lines were closer to 20 hours. That's really striking. We can rescue that particular defect by treating with even just 0.5 micromolar of longdazin, and then 1 micromolar just puts us at the same as neurotypical. So we think this is a really good start to the process of trying to screen for compounds that can shift this period length and potentially could be used to treat the sleep problems in PWS and hopefully in Hao-Fountain as well. And I'm going to show you why I think Hao-Fountain in a second here. So do anybody want to ask me up to this before I talk about Hao-Fountain? I'm not going to talk that much about Hao-Fountain, but are there any other questions about what we did and how we did it?
Yeah, I have a question. How does the longdazin work? What's the mechanism?
Yeah, I should know the answer to that. And I did at one point. I can't answer that, but it's known. I don't remember. I'm kind of agnostic when it comes to individual drug mechanisms because I want to find as many as possible. So I'm not really focused on that part yet. I'm still trying to find drugs that actually shift my pattern. But I know that drug has known function. I just can't remember what it is.
Larry, do you mind if I ask? I keep forgetting if I've ever seen this. If you put the same line in here, like on different days, PWS 148, do you always get exactly the same period? Is it as low of a standard deviation as it appears here when you have, I guess, like 4 wells in the same plate on the same day?
I think there is some variance there, and I think the variance in some of the other lines got in the way of that long versus short phenotype, because it really was just variance. But that line in particular behaves well. So you're right. We need to tease out the lines that are going to be most useful for screening, and I think 148 is one of those lines. Now, ideally, and I think we're going to be able to do this with Hao-Fountain, we get some clinical data on sleep, and we find some information about, which individuals had sleep problems versus which didn't, because we never asked, right? So for the lines we have that are Prader-Willi lines, we don't know, and we could be testing lines where those kids don't have sleep problems.
And let's say that you get all 20 teeth from that kid, 148. You think the other teeth would be
Yeah,
the same?
I don't think that's an issue at all. Yeah, the
Okay.
DPSCs are going to be the same. I don't think that's the issue. I think the issue is, phenotypic presentation, and also which individuals are clinically having sleep problems, right? So we don't know that for the PWS lines, but like I said, Christian Schaff has data on sleep for those individuals. So we feel like we're going to be able to do that for the Hao-Fountain samples more than the PWS samples. Okay, let's look at Hao-Fountain real quick. So, all right. I don't think I need to go over the features of Hao-Fountain syndrome as much for this group, but let's just generally say there's a lot of overlap with PWS and SYS. And then there's some things that are really unique, like hyperphagia in PWS and neonatal contractures in SYS. So the unique thing about MAGEL2, right, is that there are these mutations that are truncating mutations in the MAGEL2 protein. And that's really interesting in terms of mechanism to me, because as we look at different mutations, we might see different effects. At least we're hoping, right? So we'll be able to figure out something about what the protein's doing. And as I mentioned earlier, about 70 to 80% of these individuals have sleep problems. It's not just apnea, though. It's other sleep problems as well. Disturbed sleep cycle, daytime fatigue. So this is from one of Christian's papers. I should have referenced it on here. A really nice study, I think. It's just amazing how many patients he's seen and how many mutations he's characterized. So, there doesn't seem to be a theme other than, when you have these mutations, there's a downstream stop codon and a truncated protein is being made. Although I haven't seen too many studies on that actual protein, I'd like to know if anybody's been able to see these truncated proteins. And I suspect this might be the right group to ask that question to. But to me, as a geneticist, these are really interesting dominant mutations, right? Because now you're basically putting the wrench in the works, right? So you've got a protein that now, is not functioning normal because of it's binding to its normal binding partners, supposedly, and then not doing what it's supposed to do, right? Or sequestering those binding partners. So I think that's a really interesting protein to study. So what happens with circadian rhythm in SYS neurons? So this didn't make it into the paper, largely because we had 2 lines. So it was really difficult for us to make a firm conclusion. But I don't think it's difficult to see that the circadian rhythm is, let's just call it messed up, right? So you look at the control on the left versus SYS on the right, you see that not only is there a decreased period length here, but it's more importantly disrupted, right? Even the same line, right? The red lines are from the same cell line in multiple trials. They don't really show consistent patterning, right? they change from experiment to experiment. That's pretty dramatic. So we need more of these lines to be able to really figure out what's going on here. But I'm confident that this is going to be a very interesting phenotype to look at in DPSCs. Okay, so that brings us back to the figure. And so really the long and short of it is we think MAGEL2 is a key player here, which is sort of fine-tuning or regulating the circadian cycle. It's not directly binding Cry1 as far as I can tell from the literature, although you can school me on that because I'd like to know. But what we're trying to do now and what we're developing in the lab is a Cry1 assay in a different cell line for us to be able to determine which MAGEL2 mutations have what effect on Cry1 stability. We're doing that using a line that we got from another lab that is a Cry1. Actually, there's a couple lines. There's a Cry1 GFP, a Cry1 RFP, and a Cry1 luciferase fusion protein. And we're putting those in our Lumicycle or our BioSpa system and in the plate reader and trying to figure out what the normal cycling pattern is for Cry1. And then we're going to try and put MAGEL2 mutations into the system and see how it destabilizes or stabilizes Cry1 protein. It should change that pattern. Okay, so I hope I convinced you dental pulp stem cells are a really viable alternative to iPSCs and other types of stem cells even, mainly because they're easy to get, they're easy to grow, not so easy to differentiate, but they're easy to grow, and we can get them from a wider range of individuals, which to me as a geneticist is more important because you look at one individual and you make one iPSC line from that individual, you really haven't looked at the whole broad spectrum of what happens in that syndrome. So, cellular and molecular studies that we've been doing have revealed aspects of underlying disease like Prader-Willi. I didn't talk about all of the molecular studies we've been doing in Prader-Willi syndrome, but I mentioned that we did some single cell work. We have some other exciting stuff going on as well in terms of rescue studies. And Hao-Fountain syndrome, we're still really just at the tip of the iceberg, but I'm really hoping to focus more on the autism phenotypes in Hao-Fountain as well as Prader-Willi UPD and the circadian rhythm phenotype. So these are the areas that we're really focused. And I didn't even show you the autism data, which is published. If you look up, Victor is the first author, my former graduate student. Okay, so understanding how USP7 mutations work in the context of that model I showed you is going to also be really interesting to our lab. So once we haven't really put any USP7 DPSC lines in, we only have a few, but we could put PER2 into those lines and look at the circadian rhythm and see what's going on in terms of circadian defects and USP7 mutations. So, and I'm also, working all angles of that, right? So Cry1, MAGEL2, USP7, that interaction that goes on during the circadian cycle is something that we're interested in looking at. And really, we're just kind of scratching the surface. I mean, even though I'm just one lab, right? And it took us years to do this, but we've collected over 200 DPSC lines. I think we can collect a lot more. If we can continue to collect remotely, we can collect a lot more lines from a lot more syndromes. But more importantly, I think if you look through the literature, we have 46 Prader-Willi lines. Like not a lot of labs can look at that many individuals for any given thing they're looking at, right? Besides maybe DNA. But it's really, kind of important that we can even differentiate these cells into different tissue types, right? We haven't done it as much, but we've made chondrocytes and adipocytes in the lab. So once we have the stem cells, we can do a lot of things. This is just a picture of all the publications related to dental pulp stem cells that we've published since looks like 2017. So it's been a while, and more are coming. There's a couple right now that are well, at least one that's right now in review that I hope will be out soon. And this is a picture of my current lab. The stem cell guy right now is Taylor Hedgecock, but So he's the guy on the left. The guy on the right is new to my lab. He's a postdoc, Tyler. Yeah, like that was smart, right? I have a Taylor and a Tyler, so that's always confusing. So Tyler is a postdoc who's also working on Prader-Willi DPSCs, and I'm pointing both of them out specifically so that you can say hi to them at the Prader-Willi meeting, because they will both be there presenting different aspects of what we work on. And then Ben in the middle is my fly guy who is working on other projects we didn't talk about today in Drosophila. All right, and with that, I'm happy to take questions. Hope I can field a few of them today.
Yeah, Larry, thank you for your presentation today. And I was curious, two questions. One, how many samples have you received so far to get the 2 lines that you currently have? And then where do the parents request the shipping package for the samples to send in?
Right, so good question. I don't remember specifically with USP7, but in general, we have a pretty good hit rate at growing stem cells from teeth. So I'd say we're probably around 75-80% of the time we can grow stem cells from the tooth. It depends, the tooth has to be in the media within about 30 minutes. If you can get it into the media within about 30 minutes, we usually do okay. Also, some teeth look better than others. If you got one that looks really solid, usually we break it open and the stem cells are great, but sometimes they'll be like, just kind of too small or, they just aren't. And that's just random. But I think, like I said, 75% or 80% is our hit rate usually. Now, your other question was how do the parents get involved? And unfortunately, I have to slug through my IRB protocol again today because they have endless questions. It was in place for a good 10, 15 years at UTHSC. Then I moved here, and there's all kinds of questions that are new questions that the other IRB didn't ask. So I at the same time, I'm trying to make it easier on the families. So I'm doing it now with Qualtrics. So hopefully you'll be able to just click a link and then fill out the consent form online and drop your genetics report as an image. And all that'll be automatic. And you'll sign the consent, send me the genetics report, and I'll send you a kit. I'll have your address as well. So hopefully that'll be streamlined soon. And that's really all we need, right? Is like the parent support groups are going to advertise the studies, and the families can just click the link and consent, and we'll send you a kit.
Okay, so so we're just waiting for you to get that together and then we can do it. But as of right now, there's no.
Until it's approved, I can't do it. I
Okay.
can't send kits out. So I've been,
Okay.
trust me, I'm trying. I'm gonna work on it today. I promise.
It's all good. Thank you for being here.
Oh yeah, thanks for having me.
I have just, I think, 2 questions. One, I know I sent teeth for my daughter. I can't remember how many I sent, but it was a few. So I don't know if she's one of the lines or not. It was a while ago, obviously when I sent them.
I'm gonna bet that she is one because, one of them must have worked, right? So
Right.
I can't say. I don't have any of the records in front of me.
Okay. And then my other question was going back to the one slide where you show that Major 2 does the fine tuning and then USP7 does the stabilizing. So the stabilizing, so if that is not working, I'm assuming that there's a likelihood that that would be a cause for the sleep issue. My daughter has extreme sleep issues. Like the sleep is so awful for her. So I'm kind of thinking that the hypothesis might be like something similar, that it would be a cause of those sleep issues.
So you could imagine that, in the very simple model, right? And I can't prove this at the moment, but I'm hoping that we will. Cry1 gets deubiquitinated by USP7, right? And that basically stabilizes the Cry1 protein, right? Now, if you've got mutations in USP7 and they no longer are able to deubiquitinate Cry1, now Cry1 just gets ubiquitinated, degraded, ubiquitinated, degraded, so that the whole cycle gets messed up, right? And that's gonna change your quality of sleep. That's the short story, right?
Yeah, that's what I was kind of getting from that slide. I was like, okay.
Larry, can I ask a question once we're here? So do you think what's the role of MAGEL2? Is it an adapter between USB7 and the E3 ligase? What do you mean it's making it more stable? Because
So I think that MAGEL2 is fine-tuning the system. That's my I don't have the only evidence I have for that is that there's a lot of interaction studies that show USB7 and MAGEL2 bind to each other, right? So why, right? I mean, I feel like MAGEL2, since it's involved in the circadian cycle and is cyclically regulated, right? If you look down here at the bottom, BMAL and clock, they don't bind to an E-box, but they do bind to the promoter to express MAGEL2, right? And MAGEL2 is cyclical in its expression, right? It's bound to the circadian cycle. So why would that be? Well, I mean, one reason could be that it's involved in this part, right? The stabilization of cry1 and when that starts and when that stops, or, how stable cry1 becomes, right? That part of the feedback loop is a little more subtle, I think, than the per2 cry1 part, right? I think it's probably just like a fine tuning.
as a biochemist, I really want to know the mechanism. So I wonder if there is
Yeah, I do
can we
too.
know something about interaction between MAGEL2 and cry1, for example, or do we just know that MAGEL2 interacts with USB7?
I was thinking of these, like, I think it's Rachel Warwick's work, right? Like the in vitro protein binding assays that she did. And I don't recall if anyone has shown direct binding to cry1. Does anybody know? I was kind of asking that before.
Yeah, I don't know from the top of my head. I don't remember. And I was looking at her papers as well, whether they provided some, I think they started with bio-ID, identification of binding partners, and then did some IP of overexpressed proteins. But it could be like, if I think of its potential function that in Ryan's lab, we sort of figured it out, is one of this regulation of WASH, where both proteins are important in a way that maybe stabilization of or ubiquitination of USB7 can be also, I don't know, is USB7 ubiquitinated? And whether this could be like MAGEL2 promotes ubiquitination of USB7, and USB7 then deubiquitinates, like, this cycle.
Yeah.
Very typical for USB7. Usually works with E3 ligases. And as you said, this is kind of similar to what you guys saw in the WASH pathway, where different E3 TRIM27, right?
Yeah.
But they all work together as a triple complex. So something like that can probably happen here. I'm asking because we've been looking at direct interactions between USB7 and MAGEL2 lately, introducing patient mutations and trying to narrow down how they do interact. So I was wondering, and there are some mutations that actually break the interaction between USB7 and MAGEL2. So it'd be really nice to go into cells and check if that makes an effect there somehow, because we know specific mutations, we know specific sites that are important for this interaction between the two proteins.
So can you share that information with me? Send me an email. We can talk about, making constructs for the cry1 experiments, I think would be great.
Yeah, because right now, I think just in general, there's a bit of a problem with having a good cell system for studying USB7, because we don't really know what the good readout is. USB7 has so many substrates. It does so many things in the cell. So we don't really have a very good, robust readout. So
and that's exactly why I was developing the cry1 system, right? So it may not be everything USB7 does. It's very specific for the circadian cycle. But if we can change it with a mutation in USB7, we're golden, right? We're in. So
okay, let's keep in touch.
yeah, please.
It was a really great talk. I have a question that's more just out of curiosity's sake, but since you have these DPSCs coming from different patients with different mutations, do you see differences in the neuronal structure after you differentiate, or in like the ratio of the different components?
We haven't looked at
or-
neuronal structure. We haven't done that much in terms of staining. But I can, I didn't show any of the autism data, but that's a good example where, let me summarize that study quickly. So it turns out that, we had Prader-Willi UPD cases that we could split by using the SCQ, which is an assessment for autism. So we had Prader-Willi UPD plus ASD, Prader-Willi UPD minus ASD. When we did the molecular studies of those neuronal cultures, we found a huge list of mitochondrial genes that were downregulated in the plus ASD group, but not the minus ASD group. So we went and looked at the cells, and we used a marker TOM20 for mitochondria. And there were these clumps of mitochondria in the neurons, and even mitochondria stuck in clumps down the processes. So we ended up using a Maris to quantify this difference in the plus and minus ASD group, and we found a significant increase in these inclusions, basically a decrease in mitochondrial volume in the plus ASD group. So it seemed to be characteristic not only of Prader-Willi UPD, but Prader-Willi UPD plus ASD, right? Which I don't know if you know this, but they have a higher incidence. They have like a 30% incidence of ASD, higher than typical populations, right? Just the UPD group. So we've been branching out, and we've used several other DPSC lines from our collection where we have a positive ASD by SCQ. And lo and behold, they have mitochondrial defects too. So we're in the process of trying to build this story based on syndromic ASD and mitochondrial defects. And we're using some other constructs from a collaborator's lab soon, I hope, that will be more specific for mitochondrial volume, mitochondrial function, and mitochondrial health. So he has this really cool construct that we want to get in that will give us all this information at once instead of looking at the TOM20 staining and trying to figure out what's going on. So, but yeah, they can be different from line to line. Typically, the things that we worry about that are different are like quality of growing the cells or how long it takes to grow them. That's the stuff you notice. But at the molecular level, I think I showed you, if you looked at the Schaaf-Yang line that I showed you, those were different. Like the blue line, right, is 274. That individual doesn't really have a huge defect. It looks very controlled. Their circadian cycle is pretty good. It's this other line that's completely disrupted, right? That's just a mess. And you don't know these things until you do the downstream assay, right? So in terms of circadian rhythm, I would say one looks closer to control and one individual looks like they probably have sleep problems.
Hey, Larry. Yeah, it's always exciting to see your work and resource you're building. So I'll definitely email you now for potential collaboration.
Yeah.
And I think it's great, right? What we do is more exploratory to find even potential some novel mechanisms, and then going back to your models again for verification, patient samples, that would be really, really helpful. My question would be I have two questions. One is, you have done sequencing on these lines, right?
Yes.
And also you said even single cell sequencing.
Yes.
Are you building a repository on these for like maybe at least for the community, or is this published, or?
Yeah, so the RNA-seq that's not single cell is published and is in
It's available, the data source?
Oh yeah, it's in Geo, right?
Okay.
To publish it, we have to put it in Geo, so it's in there. The single cell data is unpublished because we felt we needed to, there were technical reasons. We used parse instead of 10x for a variety of reasons. We got sort of stuck in the analysis step. We did analyze it and we believe the results, but it's been difficult to convince people that our UMAPs don't look like the UMAPs that they're used to, so they get all bent out of shape, right? Reviewers get bent out of shape. So what we want to do is now do some RNA scope to validate the things that we think are being differentially expressed in particular cell types in the culture. So if we can do RNA scope with maybe 100 of these constructs, we can show what we think is already true in the single cell data, but I think more convincingly that, for example, the things we think are down in neurons that look like they're excitatory neurons, right? In PWS.
That could be something we could even work together because we have single cell data on mice. We could maybe cross-validate between species and this would be very helpful also for potential validation of the mouse model for some findings. So definitely interested in doing that. And
if once we get so it's not on the back burner, but hasn't been the front thing to do. But now that we got this other paper out the door, I think we're going to try and get a list together of transcripts, and I'm happy to share with you. Actually, remind me in the email.
Yeah, I will email you, and then
we wrote up the whole single cell paper. It's written up, and you can
Perfect.
look at the paper and see if any of those genes overlap with what you're saying, right? That's fine.
That could be something too. And I also wanted to ask, have you ever considered differentiating these neurons in these iPSCs to other cell types?
Yeah, we've published that too.
Type?
Yeah. Well, we haven't done astrocytes, but we've made chondrocytes and adipocytes and published that.
No, I was more thinking within hypothalamic realm.
That's a nice thought.
Yeah.
But in practice, I don't think anybody's actually made hypothalamic neurons in culture. There's one lab that claimed they did it, but we've tried it, and you remember Caitlin, and Caitlin tried it a few times.
Yeah.
It really
I was even thinking
work.
maybe even if not hypothalamic neurons, but maybe astrocytes or microglia or.
I think that would be useful. But at the moment, I'm kind of taking this holistic approach. Brains are made up of lots of different cells, and we always thought these cultures were mixed cultures. We always said that. And then we did single cell, and it looks like they're mixed cultures. So why beat on a dead horse, right? We've got all the right things in the pot. We're making gumbo, as they say here in New Orleans, right? Like we must see what's going on in the pot,
No, it's just interesting. I can tell you a little bit of insights from our finding is, although MHL2 is predominantly expressed in neurons, it is expressed also in other cell types. And the differentially expressed genes, with the number of them, it's actually higher in non-neuronal cells. That's why and can be extrinsic as well, right? That was just interesting, and the cell-cell communication is for sure very important. So. Yeah, thank you. So I'm looking forward to discuss more.
I just have one question in terms of if you compared your dental pulp derived neurons compared to SY5Y cells, is there differences? Obviously there's limited fidelity in electrophysiology in the Shishi cells.
Yeah.
I'm wondering if it's the same thing with your derived neurons.
So the last paper that we published with Julio's lab, they did electrophys on our DPSC neurons to look at piezo2. They were specifically looking at piezo2 currents. So it is completely possible, but not easy. Nothing's
and in
really
terms of synaptic markers like V-gluten, V-GAT, you haven't looked into those?
I mean, we haven't dug too far into the markers, but we're seeing them in the single cell data. So, we're not concerned that they're not neurons, and other people have shown currents, right? And we've shown currents. Yeah. So I feel like, this is not my area of expertise, so I don't really pursue it. But it's there.
Yeah, no, I was just trying to see how comparable it would be to like the in vivo conditions. Yep, that makes sense.
And we're actually helping my collaborators lab with sci-fis at the moment. So we are going to at least look at the mitochondrial function in the sci-fis versus the DPSEs, but not like sequencing the lines.
Okay.
Okay. Well, I hope I get emails from Clementina, who I know will email me actually, and and Irana. I hope to hear from you because I'd love to hear about these mutations. I think, this is very productive for me to see what everybody's doing with MAGEL2 and USP7.
Awesome. Yeah. Thanks.
Who's
Bye.
who's running the show? Yeah.
Thank you so much.
Okay. So, you guys do this every month or what? Yeah. So
Yes.
keep me in the loop. Let me know, and I definitely try and get on when I can get on because obviously I know some of these people already, and we already kind of work in the same areas, so it's always useful to have these conversations. And Amber, I'm going to let you know the second I can collect DPSEs again. I'm trying.
Very exciting. We are caught up on our end to keep you moving along.
Okay.
Have a great day. Thank you, everybody.
Thank
Bye.
you. Bye.